Around with this site. It's a little different than the one listed in the PowerPoint if you're looking at that. They reorganized their domain and this is now the correct site. At the moment, I think it is getting absolutely slammed, and I was in there for a minute and then I tried to change pages and it just locked me out and the whole server went down. So I guess I asked too much of it. Anyway, it's a really nice thing. You can compare the evolution rate of different viruses, and it's got the novel coronavirus and Ebola and all these flu viruses. And you can see how the genetic changes map onto where the virus is, like in the world, and also according to time. So you can see the virus changing as it's moving, and certain strains do really well or poorly. Really interesting tool, and I don't know, give it a try. See if you can get it to work, because it was working a little earlier. Anyway, meanwhile, sorry, let's go over here. So this is a model of the surface of the influenza HA, hemagglutinin protein, and it is showing the amino acids that change during an outbreak. And so as you can see, the parts of this that change are going to be the surface. Now, what's the relevance here? Well, the relevance is we didn't have a really good map of the Ebola virus surface glycoprotein, and so I haven't seen the same picture, but the same thing is almost certainly happening. So it's not the core of the glycoproteins that changes, because that's the bit with all that machinery. That's the bit with your fusion peptide and heptide repeat 1 and 2, and all the coiled springs that have to store up a bunch of energy. It's difficult to just randomly change those and still have a working glycoprotein that can go through all the metastable states. The thing that changes tends to be surface loops, and so here are all amino acids that change, and you can see they're all on the surface, and some of them are even in these sort of floppy loops that just hang out here and don't even play a direct role in binding the receptors. So that's the case. That's how Ebola and pretty much any virus changes as it's evolving. You'll get immunological selection. So antibodies that are hitting the outside and slowing down the virus are going to kill off any viruses they can bind, and the only viruses that survive are the ones that are different. And so that's how we end up driving the evolution of viruses. They would probably stay the same if they could otherwise. So one of these antibodies was part of a cocktail. It was called Zmapp. And this was a thing that they were touting early on in the Ebola outbreak. This was going to save the day. And what Zmapp is 3 monoclonal antibodies. And so shown here are 3 monoclonal antibodies, purple, red, and yellow. And they're just showing the, it's going to be called the FAB region of these antibodies, if you're into antibody things. All an antibody does is it sticks all over the surface of something that is trying to hurt you. And in this case, the antibodies stick all over the surface of the Ebola virus glycoprotein. And shown here is a nice model of the Ebola virus glycoprotein with all 3 of these things stuck to it. If any one of these things manages to hit Ebola virus, then it knocks out that one glycoprotein. But again, you've got something like 100 glycoproteins to, I don't know, maybe 300 glycoproteins on the surface of a virion. And so you have to hit most or possibly even all of those to actually have an effect. So they tried treating monkeys with this, and what they found was that if you treat monkeys every time when there is an arrow, there, and you give them Ebola virus at different time points, but always after they've been treated, then what you see is that the untreated ones are the pink, and so they make it out to 7 days and then they just die. So Ebola causes a very different disease in the kinds of monkeys they were using for this study. It just kills them right away, whereas in people we sort of linger for about a month. The same way we do with the novel coronavirus, and with a lot of viruses for that matter. But you can see with a lot of treatment, and treatment 24 or 48 hours before the virus went in there, then, oh, sorry, no, this is the treatment schedule. The virus goes in first. So, pardon me, the virus is the red arrow. And then these are the treatment schedule. And, yeah, they were able to protect most, but not all of the monkeys. So when the line steps down, that means probably one of the monkeys out of a group of 4 has died. And so you see we're right at 75%. So that's, yeah, probably what happened there. So they were okay in monkeys, and we fast forwarded them for use in humans, and it turned out they didn't work. Here's another thing that we tried along the same pathway. And so this is what happens when there's any new outbreak. People just try all the things they've been wanting to try for ages. And these are all things that protected animals of some sort in laboratory tests. And so the second vaccine, the first vaccine rather to be tried, was made by Oxford University in conjunction with GlaxoSmithKline. And what it was an adenovirus. So this is a kind of DNA virus that's really cool. It looks kind of like the Sputnik space probe. And inside of this is double-stranded DNA. And what you can do is you can hollow out the inside. So instead of carrying the virus in there, you can put a new gene in place of the virus. You just have to have a little bit at the front end and back end so it'll get picked up into this thing. And so you can turn the virus into essentially a little truck that will carry a gene from the laboratory into somebody's cells. And so it takes the gene in there, it drops it off, the gene goes into the nucleus, as happens with HIV. And then once the gene is in the nucleus, this will be in the form of a plasmid, so it can just be replicated in the nucleus, doesn't have to integrate, and it'll just start making Ebola virus proteins. Now, they're not going to make all the Ebola virus proteins, because all the proteins plus the genome equals Ebola virus, but they will make just the glycoprotein. And so they tried this out, and it turns out it didn't work well enough. They didn't get enough of an immune response out of people, and so the trial was discontinued. It was not successful. And this is very similar to a couple of the strategies that are currently being tried for coronavirus. And if you're watching this in the future, I don't know, tell me, did they work? My money is on DNA vaccination being not quite strong enough to get a good protective immune response. But you got to try it. Otherwise, you don't know. One of these days, it's going to work. So remember in the last slide set, I was telling you that there were real differences in the amount of virus in people that lived versus died. So here's a little chart that shows a couple of correlations between people who survive or don't. And so what we're going to do is we're going to look at the reason behind these differences. And there are a couple different reasons, but they all link together. They all have to do with the immune system and the way it works. So in here in the middle bar, let's just hit that one first. 10 to the 10th genomes per milliliter in the blood is a ridiculous number. So 10 to the 9th is a billion copies. This is 10 billion copies per milliliter. Crazy. In people who are going to die, but only 10 to the 7th per mil in people that are going to survive. So 10 million viruses is totally sustainable per milliliter in all of your blood. But 10 billion viruses per milliliter is just too much. Yeah. So it's amazing the body can stand up to this and bounce back. Big difference is the CD8. So these are the killer T cells. And those killer T cells, you'll have a very strong response that is going to be specific to Ebola virus. So they're going to recognize Ebola infected cells and they're going to kill them in people that survive. In people that die, their CD8, their killer T cells, do not kill the virus. They just never learn. And so it's like a T cell education problem that seems to be the thing. So going along with this, so high lymphocyte count means you have lots of both T cells and other kinds of white blood cells in your body. So basically, your body knows it's infected and it's making white blood cells to fight it off. Those people survive. Low lymphocytes generally means that your body thinks it's okay, or that it's not like a virus or a bacterium. It's not making an immune response, and that leads to death. Early antibody response tends to go with survival. No antibodies by the time you start to get disease indicates you're probably going to die, because antibodies are made sort of early, but then they slowly build up. It takes about a week to start to get a good antibody response. And the last big indicator is, so this NO is a thing called nitric oxide, and it's released by macrophages, and it can break in half, and each of those halves will be a free radical. And so then the next thing they bump into, they will just destroy chemically. And so if you get a lot of these aggravated activated macrophages, they'll release a lot of nitric oxide. and it just non-specifically destroys the place where they are. Macrophages live in your bloodstream, and so it's just corroding the inside of the veins and arteries and all the big organs. Like the liver tends to fail, the kidneys tend to fail, can even cause heart failure. And it's just because, yeah, all these macrophages are being activated at once. Low nitric oxide means you have relatively low amount of macrophage activation. You don't want zero, but you don't want too much. And that seems to correlate with survival. So summing it up here, you've got two paths. You can make ineffective immunity. So in that case, you're going to turn on your innate immunity and you're going to leave it on. Innate immunity is things like macrophages and neutrophils and just all the granulocytes, they're called. They're part of the immune system that you have from the time you're a baby. And they can recognize danger in a general sense, but they tend to overreact and they are not specific to any one type of danger. So they're kind of limited. And so in this case, what's going to happen is that your macrophages, which are one of these big immune cells we'll talk about more in a minute, are going to get infected with Ebola virus, which is terrible. And that makes them just dump out all of their chemicals, cytokines and nitric oxide and everything they've been storing up to fight against the virus. Because of that, you get damage to the veins leak, you get fluid leaking out, which is called edema. This fluid builds up in little pockets and other organs where it shouldn't be. And all of this vein leakage and fluid leakage leads to multiple organ failure, which leads to death. On the other hand, if you have an effective immune response, you're going to make an adaptive immune response. So this will be things like antibodies and T cells that specifically recognize either the virus or virus-infected cells and go out and kill those cells or clean up that virus. And so this is going to start with dendritic cells, which are a cell from the immune system. They live out in the periphery, so think like fingers and toes, but really just not the central nervous system. There they're going to pick up little bits of anything that comes along, and then they will take this to a lymph node and they will present it to some other cells. They can present it directly to T cells, or they can present it to these things called helper T cells that'll then go on and present it to the cells that make antibodies or will help out the T cells. Either way, you're going to get a killer T cell response, so that's CD8 T cell response, or the killer T cells, and that leads to virus clearance. So, in about 4/5 of cases, if you know the sequence of one particular gene in your body, you can predict whether or not you're going to live or die from Ebola. And so this is a test you could do. And if you ever get onto an organ donor list, like a bone marrow donor list, they will do this test. They will sequence this little bit because this is one of the things they want to read out. And so the region is called human leukocyte antigen type B. And it's part of a more general group of proteins called the major histocompatibility complex. So HLA-B is human leukocyte. Leukocyte means white blood cell antigen. So that's like the human version of this. And we have types A, B, C, and then we have a bunch of other types that are made on immune cells. DR, DQ, such as that. But we don't need to know those. You can learn those in immunology class. And you get 2 copies of HLA-A, one from your mom, one from your dad. Same thing with HLA-B and HLA-C. And yeah, depending which HLA type B you get, you will actually do really well or really poorly. So, for example, there are some of these alleles where you had 8 patients that had the allele and all 8 of those died. And over here, the p-value calculates what are the odds that could happen, just by random chance. And the answer is about a 1% chance that would be a random occurrence. Here we got 5 out of 6, 11 out of 14, 13 out of 17. Those are bad alleles to have. So things like HLA-B15 or B67 is a pretty bad one. On the other end of the spectrum, you've got these. So HLA-B7, 11 out of 11 people who had this did not die from Ebola virus. Now, keep in mind that people could have, the right genotype and still get hit by a bus or something like that. I don't know how careful they were at sanitizing the data and getting all those out. But there could be complications as well. But everything down here toward the bottom of the list seems to be protective. Everything up toward the top of the list seems to be pretty bad. And these were just the first sort of stats that came in. I think people are working on similar papers for the new Ebola outbreaks. And yeah, we should get a better picture of what it is that actually protects a person from this. But this is the start. Your HLA allele is the start of a beautiful, very interesting story that we're going to cover. So the last thing that you need to know about Ebola virus, just going into it, is what's the vaccine that actually worked? And so here's a nice little infographic that I think was made by the same people that made the vaccine. And so the idea is that here's the glycoprotein on the outside, and here's the glycoprotein gene in this sort of orangey, pinky, reddy color in the middle. And what they did was they took out the glycoprotein gene and put it into a different virus. Now this virus is a thing called Vesicular stomatitis virus, VSV. And it's a virus that can grow in people but doesn't usually cause disease in people. And it also, if I remember right, grows in horses and does cause disease in horses. And so they replaced the glycoprotein that's normally on the outside of vesicular stomatitis virus, this yellow one, with the glycoprotein that's on the outside of Ebola virus. So it's a fully competent virus. It can replicate and everything, but instead of having the normal outside coating, it now has the Ebola virus outside protein. And it still works for fusion and entry. And you'd look at this thing and say, wow, that's going to kill people. That's really bad. But it turns out it's not the glycoprotein that actually makes Ebola virus dangerous. It's the combination of all the genes working together that together get the job done. And yeah, swapping out one part like this is actually pretty safe. And so this is the vaccine that worked. They gave this to thousands of people, and it was very effective at preventing the spread of Ebola. In the first paper, they had given it to something like 5,000 people, and they didn't have a single infection in that group. I think subsequent papers, they found a couple of infections because everybody's immune system is different. Nobody reacts to a vaccine exactly the same way. But yeah, it was a very positive thing, and this is how they did it. So that is the end of the introductory parts of Ebola virus, and now we're going to go on to the actual immunology and virology of how this thing works. And I think you'll find that quite interesting. So let's go see.